All right, let's go ahead and get started. Two quick announcements. One of them just based off something I was just said a moment ago. For those of you who still need a health ID, the point person that'll help you with that is Karen Sleezer. She's up in the BME office on the second floor here. If you reach out to her, she can help you coordinate with you to help you get a health ID as soon as possible. And for those of you that work to get one, it sounds like some of them are already up there waiting for you up in the BME office. Second announcement is we have our first assignment due on Tuesday. Make sure my recording is working. Forgive me. Our first assignment is due on Tuesday. That's going to be a brief description on the order of a few sentences of you identifying what the disease or biological problem your project is going to be focused on. And again, to help make your life a little easier, it's good to think about identifying a project where you can either focus on, if there's not a known mechanism for the disease, you can focus on proposing on identifying a new mechanism. Or if there is a known mechanism, if there are therapies, but if the therapies are not great and they need to be worked on, you can propose an alternative therapy. But the key thing is making sure that all of this is from a cellular and molecular biology approach. Again, if you need feedback, all of the teaching team has their office hours available and posted. And if you're having a hard time thinking of some ideas, or if you're looking for inspiration, one great idea is to try and look at just the BME research page to see what are the different research questions that labs in the department are pursuing. Not every lab is going to be cellular and molecular biology focused, but a decent amount of them are. And so that might be a good way to kind of peruse through what's the ongoing research in the department that might help provide some inspiration of what your project or what your biological problem can be focused on. So for today, we're going to be focusing on enzymes. So a lot of the work that we've done so far has been first looking at amino acids and proteins, and then looking at how those amino acids and proteins help with cell structure, and particularly the cell membrane. We've been looking at those amino acids and proteins in the context of the extracellular matrix. And now we're going to be focusing on a specific class of proteins called enzymes. And we're going to be applying that within the context of botulism. So botulism or botulism poisoning is typically caused by a bacterium called Clostridium botulinum. It's a spore-forming bacteria. It's typically found it's a common soil bacterium, but there are also some key other contexts in which this bacterium can be discovered. When we say it's spore-forming, typically that means that it colonizes into a structure that helps protect it from the external environment, trying to promote its survival. But beyond common soil bacterium, they can also be found in improperly stored food. Additionally, it can also be found in improperly cleaned wound beds. But there's one other key area in which this bacterium can be found, and it's a food product. Is anyone particularly thinking about it being a spore-forming bacterium? Can anyone think of a food product that is at danger of having these spores? Yes, honey. That's exactly right. Particularly local honey. Local honey doesn't go through some of the same processes, and so that's a food product that is at risk of having these bacterium. However, that is usually only a risk factor, particularly for younger individuals on the order of being younger than 1. And so usually for anyone under 1, it's told that they should not have any honey, but also particularly pregnant people should not have any local honey, just because it's more at risk of having this bacterium. Some of the symptoms associated with botulism poisoning: flaccid paralysis, that's loss of motor neuron function and loss of muscle control. Because of that, the downstream consequences are typically patients will have difficulty swallowing or speaking. They'll have subsequent facial weakness, as depicted here in the image on the slide. They'll have drooping eyelids, dyspnea. Does anyone know what dyspnea means? Yeah. Sorry, I couldn't hear you. Difficulty breathing, exactly right. And typically that dyspnea, if it's severe enough, can be life-threatening. But some of the other more common symptoms are also nausea, vomiting, and abdominal cramps. So botulism poisoning or botulism is not very common. There's only on order of hundreds of cases annually in the US. However, despite that, I should say it particularly works, the bacterium releases a toxin called botulinum toxin. And in case any of you are thinking this sounds familiar, yes, this is exactly what's used in Botox, but at sublethal doses. And there's not very, I tried looking up the case numbers, there's not very many instances of botulinum poisoning as a result of Botox. But despite that being the case, it's still important to note that this is among the world's most deadly toxins out there. And one of the ways in which we define how lethal a drug or toxin can be is by determining its LD50. Does anyone want to describe what LD50 is? Yeah.
I think it's like the amount of needed to
Exactly right. It's the lethal dose to kill 50% of the population. And in this case, for the botulinum toxin here, it's 100 nanograms. To give you some context, that LD50 is for an average person, I think they calculate it for a 100 kilogram person. That is 4,000 times higher than cyanide. And so the reason why we're talking about this is because this toxin is an enzyme. And so we're going to come back to this toxin towards the very end of the lecture. But now we're going to pivot to talk about enzyme and enzyme kinetics. As was discussed in the pre-lecture, we're going to first discuss the Michaelis-Menten kinetic model. What I first want to say is that, as the name implies, that this is a model. All models are not perfect, and they all are designed with assumptions. And so we're going to get at those assumptions towards the end. But first, we're going to set the stage for what this does. And so this is meant to model enzyme kinetics. So enzymes are a class of proteins. And they're a class of proteins where they are meant to try and catalyze a reaction. They're meant to accelerate a reaction to occur beyond what would normally happen typically in the absence of this enzyme. And so the Michaelis-Menten model, what it does is it tries to show where the saturation or the quickest speed at which these reactions would occur at high substrate concentrations. And so the way enzymes work is that an enzyme acts on a substrate to produce a product. And so you'll have a moment where you'll have the enzyme, and the enzyme will find its substrate, what it typically reacts with, to catalyze the reaction to produce some sort of product. And so what the model here does is we have on the x-axis substrate concentration, what the enzyme targets to catalyze a reaction to produce some sort of product at the end. And so to try and orient ourselves around these graphs, we have our Vmax. Vmax is the reaction rate at which saturation occurs, like kind of where it plateaus and reaches the fastest possible reaction speed. And that's typically in moles per unit time. We typically, what's important to note is that when we can determine our Vmax, again, that's kind of where the reaction kind of plateaus out at. We can then determine from there the 1/2 Vmax. And so if you go down to 1/2 of the Vmax and look at where the substrate concentration is at that 1/2 Vmax, that substrate concentration is called our Michaelis constant. And so with knowing our Vmax and with knowing our Michaelis constant, you can use this model to try and determine what the rate of reaction is at some given substrate concentration. And so what's important to note here is that there's two key assumptions for this model that'll help you determine what the rate of reaction is. The first one is that it assumes that the enzyme substrate complex, that's the enzyme meeting its substrate, and like the concentration of that is at steady state, meaning that like the rate of change of that concentration is steady, it's zero. So it's always going to be at some level, at some steady state level. The second important assumption here is that the substrate concentration, and I should say here, substrate concentration is denoted by S in brackets. Brackets are always going to be a shorthand for concentration here. And so this assumes, the second assumption is that substrate concentration is always going to be much, much higher than the enzyme concentration. So meeting those two assumptions, then typically the Michaelis-Menten kinetic model is going to be a helpful tool in determining the rate of reaction. And what I should note here is that also the units typically for the rate of the reaction is typically molarity per unit time. And so here we're going to be able to take a moment to try and test our understanding. So imagine you're in lab, all of you are doing research for credit, and you're just trying to find, and you're working with some enzyme, and you're trying to do an enzyme rate experiment. And so given substrate concentrations for your that's being put in the presence of some enzyme that you're working with, you're given reaction rates. And so I want you all to try and take a moment to go ahead and figure out based on the substrate concentration. And if you want to, and if it helps, you can go ahead and graph this out. Go ahead and take a moment to look at the reaction rate to try and determine what your Vmax is and what the subsequent Michaelis constant is going to be approximately. Again, this is like a back of the napkin calculation or approximation. And so again, the Michaelis and remember again what we said about the Michaelis constant. It's the substrate concentration at which you're at 1/2 Vmax. Yeah, you want to go ahead? Yeah, that's it. I don't have my microphone. I need to stay back here. Yeah, 1.2, so approximately 1 is exactly right. So we can see, you can kind of visually tell that if you look at the right column for the reaction rates, you can see that it starts to plat out at around 12, which means our 1/2 Vmax is going to be 6. And so when we look at if we look at what the substrate concentration is when we have about a reaction rate of 6, it's going to be just above 1. And so that's a quick way to try and demonstrate how you can determine what the Michaelis constant is given these values. Are there any questions here on this slide? Actually, I don't want to say are there any questions. I'm going to pause and ask for 2 questions based on anything we've talked about so far. Come on, give me 2 questions. There we go. Yeah. Yeah, so the goal is to find Km, the Michaelis constant. So as we described before, the Michaelis constant, and you can kind of see this based on the graphs I've drawn here on the board. Km, that value is the substrate concentration at which we're at the 1/2 Vmax. So we can see here that the Vmax and Vmax is the maximum, the kind of the saturation reaction speed. So we can see if we look at the right column where the reaction rates are, it looks like it kind of tops out at 12. So from there, we can do, that's the Vmax. The Vmax is 12 there, just approximating. From there, 1/2 Vmax is going to be 6. So at trying to look at these reaction rates, we want to try and find a value that's about close to 6. And so there's a 5.6 there, so that's pretty close. We want to look at what's the approximate substrate concentration that corresponds to that. And so it looks like we have 5.6 reaction rate to 1. So it looks like a reaction rate at 6 will be probably just above 1. Does that make sense? And so that's just above 1. And I like the approximation you gave, 1.2. That's probably going to be our Michaelis constant for this enzyme here. Are there any other questions here? Yes. Yeah, I mean, this is like back of the napkin calculation. So like, yeah, if we're asking for a specific, we wouldn't ask you to like approximate. We'd make it easier for you to determine like and identify a specific number. Yeah. And during lecture, we're always just gonna be doing back of the napkin calculations just to try and understand the big picture. That's typically whenever we do these like little not pretend questions, these like practice questions, the goal is to help you guys understand the big picture. If you guys can understand the big picture concepts, that'll help you kind of populate the finer details of what we're talking about. But I would say first focus on like, what's the big picture? With each slide that we go through, I'm going to make sure that I do a good job of trying to communicate what's the take-home message of this slide so you guys can understand like what's the main points, so that then as you kind of go back and listen to things, you can kind of populate the finer details and really dive deep into some of these concepts. Okay, so again, an opportunity to test your understanding. I know all of you took your time to listen to the pre-lecture really intently. So based on the pre-lecture, and I should take a note to say this, we talked about Gibbs free energy a couple times already, and this was talked about in the pre-lecture. So based on these two reactions, reaction A and reaction B depicted on this graph, based on kind of the energetics here for the Gibbs free energy, which do you think will has a more likelihood of occurring? Which reaction is more easily going to occur? Yes. Yeah, B is correct. Why B? Yes, exactly right. So if you look at B, the going from left to right, the height of the hump, which is the Gibbs free energy there, the height of the hump at which it has to overcome is going to be a lot easier for B for that reaction to occur. A great way to think about this kind of, it's a bit reductive, but a great way to think about it is, if you're pushing a stone up a hill, and the reaction is the stone going over the hill to the other side, going from reactant to product, which hill is easier to overcome here? And so in this case, the answer is B. Thank you. So we're going to take a second to try and dive deep into some different classes of inhibitors. And so a lot of drugs that are currently on the market right now, like Cipro drugs or Cipro or anti-HIV drugs, are enzyme inhibitors. And so there are two major kinds of enzyme inhibitors that are typically used. We have competitive and we have non-competitive enzyme inhibitors. So the first one we're going to focus on is we're first going to focus on competitive enzyme inhibitors. A key thing to note here is that competitive inhibitors, what they do is, what we usually see is for an enzyme, when it interacts with a substrate, the substrate will bind to the active site. So on this figure here, we have our enzyme, which kind of looks like Mr. Pac-Man, and the substrate in which it interacts with, the substrate comes in and docks in on the active site. All right. And so the way in which competitive inhibitors work, and you might guess what they do based on the name, competitive inhibitors, what I should say is after the enzyme works with the substrate, it produces a product. And so that's the product there. What competitive inhibitors do is they come in and they dock and bind to the active site. And so they're competing with the substrate for the active site. And so as you can imagine, this will affect the reaction. This will affect the reaction where the substrate is going to be used to produce a product. Now, this will alter Km, the Michaelis constant here. But what's important to know is that Vmax is a characteristic of the enzyme. It's not altering anything about the enzyme. So for again, this is a bit reductive, but the enzyme is in a Mr. Pac-Man shape, for lack of a better phrase. It's still in that shape. It's still the same enzyme. It's still the same protein. Nothing is changing about it. It's still the same. And so that means Vmax is not changing. It's just what this is doing is that it's taking it longer for us to reach Vmax. And so what I've done is I've drawn a series of reactions on the board here on both sides to try and help everyone see. So the solid black line is our average reaction with enzyme X. The dashed line is in the presence of a competitive inhibitor. So what that means is that this is going to change the Km. It's going to change where the 1/2 Vmax is going to be at, but we're still eventually going to reach Vmax because that is a key quality or characteristic of our enzyme. And so to try and help spur you guys to think about this a little deeply. So if we push the substrate concentration high enough, again, I've already kind of given you the answer a little bit, but if we push the substrate concentration high enough, what will happen to V? Does anyone want to venture a guess here based on what we've already said? It's not a trick question. Kind of already given the answer. Will it reach Vmax? Yeah, exactly right. It's going to reach Vmax. I kind of scooped myself. I forgot that line was there, so forgive me. So again, the key things note here, the key takeaway is that for competitive inhibitors, they bind to the active site. All right, that's one of the key takeaways I want you to note here. And it does not change Vmax because that's a quality of the enzyme. Yes? Yes, this is a great question. So we're going to talk about non-competitive inhibitors in a second, but they're both it can be reversible. It can bind and unbind. but it can vary from inhibitor to inhibitor. So we talked about competitive inhibitors. The next class is non-competitive inhibitors. So our competitive inhibitors bind to the active site. Non-competitive inhibitors do not bind to the active site. They bind to what we sometimes call the allosteric site. Allosteric sites, or allostery in general, is this idea of working from a distance. So this non-competitive inhibitor is going to be providing inhibition, but doing so from a distance, meaning it's not actually acting on the active site directly. It's binding to another site that we typically call the allosteric site. Sometimes we call it the regulatory site, but for now, we'll call it the allosteric site. And so what will end up happening here is that we have our substrate that'll bind to enzyme X. It'll bind to the active site, and it'll produce a product. If we have a non-competitive inhibitor bind, again, it's binding to the allosteric site. It's not binding to the active site. What it does then is it's going to change the conformation of the active site. So one key thing that we're going to come back to multiple times today is that when we've talked about amino acids and we've talked about proteins, though many of these amino acids, they have a charge. Typically, it's in an environment that has a specific pH. So when we bring in another protein or another molecule, we're typically bringing something that has a charge. If we change the charge of the protein, we're changing its conformation, and in turn, we're going to change it. It's going to have an effect on its function. So here, this non-competitive inhibitor is going to come in and dock on the allosteric site. That's going to have an effect on the conformation of the protein, of the enzyme. And that's going to change its conformation down at the active site. So now that it's a semicircle shape, and the substrate was originally a triangle, is it going to dock as easily there as it did before? I'm seeing some head shaking no. You're correct. It's not going to dock as easily. So we're going to have a lower affinity. We're going to have a lower affinity for the substrate there because of this non-competitive inhibitor. Now, the key thing to note here is that with a theoretically perfect non-competitive inhibitor, the idea is that it will probably only alter Vmax, but the reality is it does that's in theoretical lab conditions, but in reality, it is going to alter both Vmax and Km. And so now, again, having this exercise, and I'm not scooping myself now, so I actually want you guys to think about this. What will happen to the reaction rate? What will happen to V if you push the substrate concentration high enough? And so when we say what will happen, talk about it in terms of the reaction rate and Vmax and Km. Does anyone want to venture a guess what will happen here? So if we push substrate concentrations high enough, and if this has a lower affinity now, are you raising your hand? I couldn't tell if you were supporting yourself or raising your hand. Go ahead. What will decrease? Yes, the Vmax will decrease. So because it has a lower affinity now, appreciate you speaking up too. Thank you. Because it's affecting and changing the active site, it's going to have a lower affinity, and that lower affinity is going to alter the ability for these reactions to occur. It's going to happen slower, and it's going to reach its peak, its saturation, at a lower Vmax than it originally did. Yes. Again, theoretically it doesn't, but in reality it does. Like we'll have a new 1/2 Vmax, and we'll have a new Michaelis constant, new Km. Are there any other? Yes. The same rate. That's the theoretically yes. Typically, no, in real life circumstances, no. Theoretically, the slope would be the same. Theoretically, that's why we say theoretically it only alters Vmax, because if the slope is the same, the 1/2 Vmax isn't changing. It's only the Vmax, but in reality, it is changing, and so we typically see a change in the Michaelis constant as well, because it's not, the slope isn't quite the same typically. It would, yes, the slope is different there, but we still reach Vmax. Looks like you have another question brewing. Okay, these are great questions, by the way. Yes. Great question. Yes, it would go back. So because that the charge associated with this non-competitive inhibitor is now going away, it's reversible. So now the active site, it'll go, the protein will go back to its original conformation, and now the active site, and I guess in this example, will go from this like semicircle shape back to the triangle shape, to which it has a better affinity for the substrate and will have a better reaction rate. Great question. All right, in the interest of time, I'm going to have one more question in the interest of time. Go ahead. Sorry, I just want to make sure I'm following correctly. Can you repeat that one more time for me? Yes. So if the way what we're describing now is with the assumption that the non-competitive inhibitor is staying there on the enzyme. In a realistic situation where a non-competitive inhibitor unbinds from the enzyme, and again, we're talking about this at the single molecule level. We typically have many non-competitive inhibitor molecules and many enzyme molecules, and so this is happening at a speed. And this is also all dependent at the affinity for the non-competitive inhibitor for the allosteric site as well. So like in a realistic situation, the idea is that if it's reversible, it'll undock, but then a new non-competitive inhibitor will come in and dock. And so if it's an effective enough non-competitive inhibitor, it will lower the Vmax. If it's not a great non-competitive inhibitor, it might, I guess the idea is that the affinity, the degree to which the inhibitor works is the degree to which it lowers the Vmax. Does that help answer your question? Okay. I love these questions. I got to keep going. So I'm going to try and not go too fast, but also leave room for questions. So we talked about the allosteric site. So these inhibitors or this regulation at the allosteric site is typically, we refer to that as allosteric regulation. Again, this is working from a distance, working on a site that's not the active site. Typically, another term that, so for the active site and the allosteric site, you'll sometimes see different names. If you want to stay up and look up at protein structures on AlphaFold or on Wikipedia, just to help give you some nomenclature, the active site is also sometimes called the catalytic site. And the allosteric site is sometimes called the regulatory site. And so in this image here, the R and C stand for regulatory site and catalytic site. And so what happens, what's important to note is that whether it's a non-competitive inhibitor or some other molecule that acts allosterically, that molecule will bind to the regulatory site, or also known as the allosteric site, and it's going to work from a distance. It's going to alter the shape of the enzyme to alter the active site. Now, this is key. We talked about how this allosteric, these changes, the allosteric changes on the active site promote inhibition. It's also possible that it enhances and elevates the reaction rate. And so it can change the active site or the catalytic site in such a way where these reaction rates speed up. And so allosteric regulation can have a positive or a negative effect. And so from this image here, you can see that, again, we have an allosteric inhibition on this image on the slide here, where we have a molecule that binds to the allosteric site, changes the structure of the catalytic site, and we have little or no product formation as a result of that. One key thing that'll help us here is we've been talking about subunits and domains, and it's easy for these things when talking about proteins to seem interchangeable, but they're actually quite different. It's important to note this. And so when we talk about allosteric domains or catalytic domains, domains are these important structural or functional sites on a single protein. So it's a single polypeptide protein. Excuse me. And these domains are typically just different regions of one single protein. When we talk about subunits, are typically two separate proteins that come together to form a macromolecular structure, macromolecular protein that work together in such a way where each protein is a subunit of this assembly and each provides some sort of function. And so to help kind of give you some bearing here and to use some language that we've already talked about, when we talk about domains, we're talking about different parts of a tertiary structure, different parts of a single polypeptide. When we talk about subunits, we're talking about quaternary structure. We're talking about multiple proteins, multiple polypeptides that are coming together to form a certain structure. And so the reason why this is important is because when we talk about the botulinum toxin, we're going to be talking about different domains that have different functions. But again, it's all part of one single protein. An important thing to note here is that you also see light chain and heavy chain above. When it was first discovered, typically there's a technique called Western blotting where we will separate proteins based on their weight, and this protein kind of separates into two chains that show up on a Western blot, and it's typically called light chain and heavy chain. You'll see that in some proteins, but what's more important to note here is that there are three distinct domains that have really key functions for how the botulinum toxin works that we're going to come back to in a second. So allosteric regulation can happen in a few different ways. We have covalent modifications. We have groups that can be added or removed allosterically to regulate the enzyme. One example is the addition of phosphate groups. And so phosphate groups are typically added on by a kinase. So again, any protein that has an ASE on the end is always an enzyme. A kinase is an enzyme. The main function of a kinase is just to add phosphate groups. That is their sole purpose in life, to just hand phosphate groups off to other proteins. So what can happen here is that an enzyme can have a phosphate group added by a kinase. So we have kinase X here that comes in and brings a phosphate group over to enzyme X. Phosphate groups have a charge. They have a negative charge. That charge is now going to change the conformation of our enzyme, of enzyme X, and that then is going to allosterically change and regulate this protein. And so that's one form of regulation here. Phosphate groups are typically added on specific amino acid groups. There's three specific amino acids that phosphate groups can be added to: serines, threonines, and tyrosines. As an example, there are specific kinases that are added to tyrosine. They're called tyrosine kinases. That'll be easy to remember. In my head, that'd be funnier coming out, but I think you guys are probably tired. And so there could also be, and I should also say, kinases add phosphate groups. Phosphatases remove phosphate groups. There's also the cleavage of bonds. And so what ends up happening is that typically any protein can have a precursor form. And we typically refer to them as the pro-protein. And the pro is just like an indication that like it's not a mature form of the protein. So here we have proenzymes. Proenzymes are kind of like an immature form of the enzyme, and they have something that's called a pro-cap. If that pro-cap is removed from the enzyme, it's mature and ready to go. So we have enzymes that cleave off the pro-cap on enzymes to make it go from a proenzyme to now a mature enzyme. One example of this is in the clotting cascade, where we have the cleavage of prothrombin to thrombin. And so if this is just a modification that can happen to enzymes that will, again, allosterically regulate them. This is happening not on the active site, but will then help it be in its mature form to have an active site that's ready to bind to its substrate. Enzymes are meant, their sole purpose in life is to find their substrate. And so the allosteric regulation can either help prepare them for that or can mess up that fated interaction. We have also the association and disassociation of specific subunits. And so one example of this is that when one protein binds to another, an example is when a receptor binds to what it's meant to bind to, its ligand. Sometimes there's two receptors that come together when that binding happens. When we have the association of those two receptors, that association will all of a sudden enable a tyrosine kinase at the bottom of that receptor to promote the phosphorylation of other proteins and promote signaling. And so the key takeaway for this one is just that this association of subunits can actually alter specific enzymes to promote their activity. The last thing is a really great concept that's important for you guys to know. It's called cooperativity. What this means is that this regulation occurs whereby when an enzyme interacts with its substrate, we get a product. That product now can become, actually promotes more product to be formed. So the product that's produced by this reaction actually promotes more product to be formed. So it's typically when we think of catalyzed reactions, we don't typically think of the product being part of the reaction anymore. In cooperativity, it actually comes back in and contributes to the formation of more product. In some cases, this can actually imply that there are multiple catalytic subunits on a protein, or in one case we're about to talk about, that the product is the substrate itself. And so that might seem really esoteric, but we'll actually talk about what that looks like in a second. Yes, do you have a question? Great, we're on the same wavelength. So cooperativity, again, let me catch up with my notes here. Cooperativity is, again, where the product that's formed by the reaction promotes more product to be formed. The way in which this is typically modeled is by something called the Hill coefficient. It's an exponent that's included within the Michaelis-Menten model, and it's something that's typically defined experimentally. What's important to note here is that I'm not going to ask you guys to try and memorize where exactly the coefficient is. I mean, it's not too complicated. I think I have faith in all of you that all of you can memorize this, but rather what I want you guys to do is kind of focus on the takeaway that the key concept with cooperativity is that the product now contributes to the reaction and that it can accelerate it. And so if you look at the Michaelis-Menten kinetics graph to the right, bottom right here, you can appreciate that as the Hill coefficient goes up, that means kind of like more cooperativity, more of the product is more able to contribute to the reaction. You'll actually see that the rate of the reaction increases pretty dramatically as the Hill coefficient goes up. One really great example of this is with a enzyme called glycogen synthase kinase 3, or GSK3. Again, with any enzyme, their names can look pretty intimidating, but if you take a second to look at its name, it will tell you what it does. So glycogen synthase kinase 3 is a kinase that phosphorylates glycogen synthase. Glycogen synthase is also an enzyme. You can probably guess what glycogen synthase do. Synthases are enzymes that typically make things. Glycogen synthase promotes the production of glycogen. So what GSK3 here does is it phosphorylates at a specific motif. Motifs are usually specific amino acids, a specific collection or array of amino acids. And so the substrate motif at which GSK3 adds phosphate groups is where there's a serine. The 3 X's mean any amino acids, and then a phosphorylated serine. So what it does is that it's going to phosphorylate that first serine. So if we look down here at the image, we have a glycogen synthase model. We have what's called a priming phosphorylation event. This happens via, I believe it's casein kinase 2. I might be wrong about that, but I'm pretty sure. That first phosphorylation event happens. And now we have a serine, the serine with the 2 under it. After this priming phosphorylation, we have serine, XXX, phosphoserine. So now GSK3 sees its motif, adds a kinase group to serine 2. Now, if you kind of shift your focus over a little bit to the left, now that phosphorylated serine, the serine at serine 2 that we just phosphorylated, that now helps create this. This is the product now. But now it becomes the substrate itself because we have another serine XXX phosphoserine. The kinase will add a phospho at serine 3. We shift over again. We have another motif that can be phosphorylated. And so this is just an example by which the product of the reaction actually is now becoming the substrate. And so this can create really fast kinetics of this reaction here. So I'm going to take a second. A key thing here for what we're going to talk about with botulism is dependent on some neuroscience, neurotransmission. Rather than me stumbling through neuroscience as someone who's not a neuroscientist and think it's a bit of a black box, we're going to watch a video here. So I'm going to try.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
All right, so the key thing that I want you to focus on here that's going to be relevant to botulism is we have these vesicles that fuse with the membrane that will enable the release of the contents of the vesicle. In the case of neurotransmission, it's releasing the neurotransmitters across the synapse. Because we have a membrane for our cells, and these vesicles have their own membrane, the ability to fuse this membrane of the vesicle with the membrane of the cell is an incredibly it's a high energy event, because again, we have to break these like incredibly high hydrophobic interactions within these membranes and fuse them together to enable us to happen. And so we're going to talk about how this happens and what botulism does to that. What a synaptic vesicle really looks like is this. It's littered with a bunch of different proteins on its surface across a phospholipid bilayer here. One of the key proteins that we're going to focus on as it relates to botulism is synaptobrevin. So if we look at how vesicles fuse here, what we have here is that in general, we have this class of membrane proteins that are called snares. Snares, what they do is that they bind to each other and they help pull vesicles and membranes together. Whether it's endocytosis, things being brought in to the cell, or exocytosis, like neurotransmitters being released, vesicles binding to membrane requires these snares. We have two species of snares that we're going to focus on. We have vesicular snares, or V snares, that come from the vesicle membrane, and we have T snares, which are the target snares that come from the cell membrane. So what ends up happening here is that, again, I talked about that this is a high energy event. What ends up happening here is that the V snare and T snare, as the vesicle comes in proximity to the cell membrane, the V snares and T snares will interact with each other and create these coil-coil interactions where there's sufficient enough energy produced physically to promote the fusion of the cell membrane and the vesicle membrane. A great way to think about this is that with this coil-coil interaction, if you think of like a big rope, like a tug of war rope, if you start twisting it, it'll all of a sudden create these like huge coils that kind of occur in the middle. That's pretty similar to what's happening here between V snare and T snare, where they will coil up like that and have sufficient enough energy to fuse these membranes together. So synaptobrevin, that protein we talked about a moment ago that's on the surface of vesicles, is a V snare. Again, V snare for vesicles. T snare, it means target, but that's the cell membrane snare, membrane protein. Synaptobrevin is a V snare on the synaptic vesicles, specifically containing neurotransmitters. What's important here to bring us back full circle, and I'm gonna get us there in like 2 minutes, so I'm gonna try and get us right here out of here at 3:15. Botulinum toxin is an enzyme that cuts synaptobrevin. And so the consequence of that is going to be talked about here. So we talked about how the botulinum toxin comes from the Clostridium botulinum bacterium. If you eat some dirty soil or eat some improperly stored food, I hope that none of you suffer that fate. What you'll have in your gut epithelium, that bacterium. What's important to note is, I think I've said this before already, this is a class where things are still being discovered about some of the things we're going to talk about. So only just a couple of years ago, the specific cell that is part of this pathogenesis was recently identified. We know that botulinum toxin crosses the gut epithelium. We talked about epithelium before. There's different kinds of epithelial cells in the gut. The specific one is called the M cell here. This isn't important for you to know, but rather illustrate that science is still identifying some of these things. What ends up happening is that this bacterium multiplies in the gut, secretes a toxin. The toxin crosses the gut epithelium across the M cells because the bacterium can't cross the tight junctions between the gut epithelium, but the toxin can. And so what ends up happening is that once it crosses the layer of cells that lines our guts, the toxin will then get into the bloodstream. Now, again, the toxin is an enzyme that cuts synaptobrevin. How do we get from the bloodstream to specifically a motor neuron? That is its fate. Its fate is to find synaptobrevin and to cut it. How do we get there? So we talked about how there's these different domains. There's a receptor binding domain in the toxin that specifically targets motor neurons. And so that membrane protein will get endocytosed. And so to the right side, we have the receptor binding domain that helps bind specifically to motor neurons and gets endocytosed, taken in by the motor neuron. Now, it's in an endosome, okay? Things that are endocytosed are brought into this vesicle that's called an endosome. Typically what's happening is that the pH is slowly going to get lowered to degrade and destroy to sometimes destroy what's been endocytosed. Now, we talked about charges affecting protein conformation. Lowering the pH means an enrichment of hydrogen ions of protons, which will change the conformation. This is kind of actually cool. This will change the conformation where the TD translocation domain will then open up and it creates a harpoon where it shoots out of the endosome and it escapes. Now, once that happens, it's in the cytoplasm of the motor neuron. We'll have dissociation of just now the catalytic domain, which will now target synaptobrevin in that motor neuron and destroy it. I got excited and I scooped myself again. So the toxin escapes the endosome, the vesicle there. And so after that happens, again, it kind of like unfolds because of lowering pH and shoots out. The light chain will separate off, and that is where the catalytic domain, the active site is that targets synaptobrevin. This light chain, the catalytic domain, this is called a zinc endopeptidase. So again, a slightly intimidating name. It requires zinc. Endo means it cuts in the middle. So it's cutting synaptobrevin in half. And once we have no synaptobrevin, we have no vesicle fusion, no neurotransmitter release, and that results in paralysis. I was going to do a calculation, but I'm going to save that for another time. Treatment, there are antitoxins, which are antibodies that target the toxin. However, that does nothing for the motor neurons that are already destroyed. There are also antibiotics to clear out the bacterium and respiratory support. If bacterial toxins or membrane fusion excites you, if you're really fascinated about that, here are a couple of professors at UVA that are studying that. Thank you for your time. Sorry for going over by 2 minutes, and I'll be happy to answer any questions up here.